everyone, my name is Isabel Lucarelli and I'm a sophomore at Southern Connecticut State University. On behalf of Dr. Edgington and all of our phage hunters at SCSU, my colleague Erica Sellers and I will be discussing our exciting research into two newly discovered cluster AZ phages, Elise and Niobe. Students in the phage discovery course at SCSU have found four closely related temperate bacteriophages belonging to the AZ cluster, Elise and Nitro in the year 2019, and OSA16 and Niobe in the year 2020. The 26 phages belonging to cluster AZ infect the host Arthropacter globiformis and have an average genome length of 43.6 kilobases and GC percent of 67.1%. Students in the bioinformatics course at SCSU then performed genomic analysis and annotation, which has led to the discovery of a putative TA system in phage Elise. A toxin-antitoxin system, or a TA system, is a set of closely linked genes that together encode for two proteins, a toxin and an antitoxin. TA systems are present in prokaryotes and are evolutionarily advantageous due to their functions, including performing programmed cell death on infected cells and promoting antibiotic resistance. There are different types of TA systems that are classified based on substrates and how they inhibit cell growth or destruction. One of these types utilizes ADP ribosylation, which is the addition of an ADP ribose to a part of a protein involved in cell signaling, DNA repair, gene regulation, and apoptosis. In this case, the antitoxin will bind to the toxin to prevent harmful ADP ribosylation of the bacterium's genome. In this way, the adjacent genes of the TA system are co-regulators. Gene 4 in Elise had a protein that contained two significant CDD domain hits, an ADP ribosyltransferase exoenzyme, an enzyme involved in ribosylation, and VIP2, a family of an actin ADP ribosylating toxin. To identify if we had a toxin, we ran the protein sequence of gene 4 through the HAMR database and got a significant hit to the Actinobacteria micromonospora echinospora. We then took that sequence hit and ran it through BLAST-P and got another significant hit to Actinobacteria pseudo-Arthropacter scissotolerans. We then hypothesized that if this is a TA system, we would expect synteny of the adjacent toxin-antitoxin among other phages and/or bacterial species. We're able to find synteny of the toxin gene among phages of different clusters and FAMs using Famerator. In FAM 55022, 23 out of 26 cluster AZ members and 3 out of 7 cluster EH members showed synteny, as well as the OR-FAM 55799 with a member from cluster EH. We also found synteny in FAM 43301 with members from clusters AO, AO1, AO2, and AO3, AR, FH, and FL. We then wanted to see if this gene conservation was also present in bacteria, and if by identifying conservation we could also infer the presence of the adjacent putative antitoxin. And we did. On the left is a motif alignment showing conservation of the antitoxin in different bacterial species and phages created by MemeSuite. The next question was whether or not these motifs were present in prophages to further demonstrate lineage. We ran the nucleotide sequence of the putative antitoxins of M. echinospora and cluster AR phage ARV1 through FASTER and got hits to regions of prophages for both species. On the right is a graphic generated by BLAST of a section of the genome of M. echinospora where we have identified the predicted toxin and antitoxin. To summarize, we were able to identify a putative TA system in Arthropacter cluster AZ phages and prove genome conservation of both predicted genes between bacteria, prophages, and phages. Possible future research would be confirming the same gene set and order in other bacterial species and whether or not prophages are present for those species. Determining the specific toxin substrate would be interesting as well. Now my colleague Erica Sellers will discuss a putative thiamine-binding protein in Arthropacter cluster AZ phages.
Hello, my name is Erica Sellers, and my project is on a putative thiamine-binding protein discovered in Arthropacter globiformis cluster AZ phages. Thiamine is necessary for all organisms as it is involved in the metabolism of glucose, and most bacteria can even synthesize it. It increases when amino acids are not abundant and when oxidative stress is put on an organism. This gene may also have a role in protein-protein interactions as it is involved in the binding of small molecule ligands. 6 out of 26 phages in the cluster AZ have this gene, although some have not been manually annotated. This gene was originally discovered in Elise last year, as it was the second reverse gene in the AZ phages, so it was looked at more closely. Alignments are shown below, and while there is a significant gap in eraser, this is thought to be due to a different start site needing to be called than the one that was automatically called. This gene was found to have conserved domains, as it is part of a CL00307 superfamily, could be part of 3 different families within that. 2 of them are unknown or uncharacterized proteins, including the TIGR00106 MTH1187 family or the COG0011 family. However, the PFAM01910 family is a thiamine-binding protein, which also has a pocket that binds an unidentified ligand and is upregulated under oxidative conditions. Comparative genomics analysis was done on this gene, and first it was compared to 14 other protein sequences with good E values from blasting. It was most closely related to Arthropactor bacterial proteins, which was interesting as it was furthest related from the viral protein that it was tested against. Even though the virus was found far away, it was expected that as a virus, it would probably be linked closer than bacterial. This comparative genomics analysis was also done on the PFAM01910 family, and it was found that the viruses branch from a common node with Bacillus anthracis and Chloroflexus aurantiacus. In summary, a putative thiamine-binding protein with a conserved domain in a superfamily of thiamine-binding proteins has been found in 6 cluster AZ phages. These are extremely similar in amino acid sequences when accounting for non-manually annotated sequences, and it's found that very few viruses have this gene other than the ones we have discovered, as only one found in Inner Mongolia, China has this, while many bacteria do. Further research needs to be done into potential protein-protein and thiamine binding sites, as well as looking into potential motives and potential uses the virus may have for this gene. We would like to thank the Howard Hughes Medical Institute, along with the SEA PHAGES program, and the Southern Connecticut State University, along with our fellow students who have been working with us on this. We'd also like to thank Dr. Graham Hatfill and Dr. Nicholas Edgington. Thank you all.